Welcome everybody. Like take just like 5 seconds to stretch in your chair, and then I'll get into some really exciting work we've done with differential abundance statistics on FTMS data. Usually when we think about differential abundance as biostatisticians, we're addressing specific omics, which, as you've noticed, FTMS is a very special kind of data. So here we're going to demonstrate how the differential abundance pipeline can be helpful to answer specific questions and get more conversation started on what we can do to push the FTMS field forward, what's appropriate for it, what's not, what can we use that we've already done in other fields. All of that I am very excited to talk to you guys about and would love some conversation about it in the Discord channel and as we meet later. So a little bit about me. I do multi-omic statistics, top-down proteomics, and metabolomics. Done a lot of R package development and Shiny application development, containerizing those so that they can live on servers, and people who can't code and can't implement Docker containers can use those in like a cloud computing setting. I am here with you today to talk about differential abundance and if you guys get to it, if you guys are interested in coding, we have a vignette this afternoon that you can go through for doing this kind of analysis. There is no GUI implementation, it's new. So that we will do later today, and I'll be happy to help you on along with my colleagues that are presenting this afternoon. And then I'll also be with you on day 5 for the student projects as your stats and data scientist experts. That's my email. I get lots of emails, feel free to add to those. And then always you can connect with me on LinkedIn. I like it when people do and stay connected and keep conversations going. So Damon, if you're here, I'm going to let you introduce yourself now.
Yeah, hi everyone, I'm Damon. I am also, like Natalie, a postmaster's research associate here at PNNL, working in the data science and biostatistics team in the computational biology group. Some of my kind of more specialties are biostatistics, statistics, R, data visualization. I got my master's at the University of Minnesota in biostatistics. Yeah, and then this is my email, damon.leach@pnnl.gov. And yeah, if you have any questions, you can also reach out to me as well.
All right, thank you, Damon. I am from the University of Oregon, so, ooh, ducks. Okay, so from Daniel, we learned about how to use Frida for FTMS exploratory data analysis. Natalie, before that, kind of showed you the pipeline from Core MS to Frida. You've processed through Frida, you've got some results, maybe you've got some new variables in that emeta file we were talking about, which is information, additional information per peaks, and now we want to use that information based on our sample groupings to ask specific questions. So that's what we're going to do here. We're going to talk about formulating a statistical question and the kind of questions where using differential abundance is appropriate. And then this evening, this afternoon, you guys have the opportunity to make your own questions and form your own analysis with the help of our experts. So should mention that figures in the slides, especially a little bit later, are from a variety of different data sets, so they might not be directly for the rattlesnake example that we have here. There is no analysis of the spruce data set in this presentation. That's for this afternoon for the students to go through. And also, I will be talking in some generalities so we can draw comparisons across what a general differential abundance or expression pipeline looks like, and then compare that to what we're going to do here and how that's unique and how this approach is unique. Just a quick recap of what we've been doing on day 1. You guys covered experimental design, sample collection, instrument runs. You'll revisit some of that on day 5 as well. From the spruce data sets on day 3 from Yuri and Will, we talked about compound identification with Core MS. And then now we're going to get through, we talked a little bit about filtering. We have some other filtering options, which we'll go through. Normalizing, why we do it, how you do it, and the many different methods you can normalize FTMS data with. And then we'll get into statistics, specifically differential abundance statistics, where we're comparing 2 groups at a time and seeing which of those peaks with those molecular assignments are either up or down expressed, which should sound very similar if you've done a lot of like metatranscriptomics work or any of the other omics, lipidomics, metabolomics. Those are things we typically look for, is specific biomolecules with a differential abundance in this case. So back to your data sets with spruce, you've got 3 temperatures at 0, 4.5, and 9 degrees Celsius, and you've got multiple depths, 10 to 20 meters, 40 to 50 meters, 75 to 100 meters. You'll notice that we don't have the 40 to 50 meters range for the 4.5 degrees Celsius, this data set. And the kind of questions you might be answering is how do soil compounds change as the ambient temperature is increased? So the ideas behind this spruce data set and the rattlesnake data set is as the ground is warmed up, we've released more accumulated carbon in the soil into the air. And as we know, CO2 in the air contributes to the greenhouse gas effect. So the relative abundance of organic or carbon-based biomolecules detected in the soil will decrease as the temperature increases. So stressed microbes tend to eat more biomolecules to survive. And the kind of questions you might want to ask is this effect linear? Do we see a linear increase in the amount of these biomolecules that are eaten up as the temperature increases? Or does this effect change at different depths? And this is sort of how you ask a specific question, and then we formulate the data in our analysis so that we can answer that question with statistical significance. So a general pipeline for differential abundance or expression is first you would format the data in the way that we've shown you before. The same people who have worked on all of these downstream statistics packages, the same people who worked on the FTMS-R analysis packages. And so a lot of the design, a lot of the functions, a lot of the things you've seen, we use these same common themes and these same common names. So an E-data file is an expression matrix where you've got each of your biomolecules, or in this case, each of your peaks across all of your samples, and then your metadata. Your F data is just additional metadata about each of those columns. How do you want to group all of your samples together to make statistical comparisons? And the E-meta is just additional information about those biomolecules, which is going to be really important for this kind of analysis where we don't end up with a specific biomolecule. We might have a molecular formula, but we don't always have an identification for that molecular formula. So then after you get your data, then you have any pre-processing. If appropriate, you may impute values. That's more common in differential expression, where we have more data just in general, and we know what the trends look like. If you impute values, especially on proteomics data types or metabolomics data types, you can change the mean of variance, which can affect when the missingness, when the truth data is not known. So it's only in certain cases where I've seen imputing, typically saved for transcriptomics data sets. Then also, you saw with Daniel's example earlier, you often have to log transform abundances, specifically mass spec abundances, because if you just plot it, you're just going to get a whole bunch of squash box plots, and you're not going to be able to see those actual differences between each of the samples. Then you get to quality control, where you would apply many filters. We saw a lot of those filtering options that are FTMS specific. You can have filters in proteomics for degenerate peptides, for example, contaminants, biomolecules only existing in one group. You can't really do statistics with an N of 1, so you're going to have to remove those biomolecules. And then you would normalize. This is where it gets interesting, because you want to introduce a normalization method that doesn't induce statistical significance where it isn't there, and also doesn't remove statistical significance that exists beforehand. We need to normalize so we can do our downstream statistics, but we don't want to actually mess up our data or introduce bias unintentionally. And so part of what Damon's going to be talking about is how we can normalize without introducing this bias into our data sets. There's a really cool procedure called spans that we use, which tries a whole bunch of normalization methods and selects the optimum one, or the one that is less likely to introduce statistical significance where it doesn't exist. So after you've got the data normalized, then you can move on to the statistics step, where based on your experiment question and your covariates, your groups that you've decided you're going to compare, you select your statistical method based on the number of overall observations. And then especially in proteomics and very much in transcriptomics, when you have a lot of things that end up significant, you're going to want to do some sort of p-value adjustment, because some of those will be significant by chance. They're actually not significant. But at the end of the day, the end goal of this entire pipeline from A to Z here is that we get differentially abundant biomolecules, or in our E-data file, each of the rows. So in the other omics, that could be like a protein or a gene. And in our data set, those are peaks. And so we want to identify those specific peaks that are differentially expressed between our groups, between whatever the condition is and the control. So when we typically think about differential expression, we're talking about transcriptomics then, where we have count data, that's RNA, and we've done some sort of sequencing on this data. So we have actual physical counts of the number of transcripts in that sample, which is different when you switch to proteomics and you're measuring proteins, typically with LCMS. This is a very broad generalization, just so we can talk about what generally happens. You would get abundance values, and those abundance values are relative. So it's not like a count when we actually know how much is there. We have an idea of how much is present. And that's typically LCMS for proteomics. Metabolomics can be GCMS, LCMS, or NMR, which in NMR, your abundance is different. That's an absorption abundance. And so I'll get into that a little bit later. And then we have our example, which, depending on who you ask, I think it's totally fair to also think of FTMS as a type of metabolomics, as a type of high-res metabolomics. But since we aren't specifically selecting with a library, let's say 4 metabolites or 4 proteins, I feel like kind of thinking of it as an any omic is appropriate. Both the technology and the data type really changes how we conduct this differential abundance and this differential expression pipeline, because as I was mentioning, each of the data types is a little bit different. Each of the technologies we use is a little bit different, even though we end up with a similar looking objects every time, some sort of expression matrix. The actual statistics we do varies depending on the technology and the data type. And so that was the point there. As we move on, this is an example of where technology has an effect on what you do statistically for differential expression or differential abundance. Sequencing, as I mentioned, that expression is actual count data that you can get. With mass spec, we're talking about intensity abundances, which are much larger values and can vary a lot between instruments, between runs, between each of your samples. And so you really do need to normalize these abundances so that we account for that natural variation that occurs between each of the instruments and each of the samples. And then NMR is also commonly used in metabolomics, and that also returns abundance values, but those are based on magnetic absorption. So for example, here, NMR data doesn't often require normalization because that absorption value you get is more indicative of an actual concentration, more so than mass spec. So this is just an example to say that like there isn't exactly one pipeline every time. There's tools that we've developed here to help you make informed decisions on your pipeline, but it's not like you can just make one and run any type of omic on it. Also, data type affects what you do when your differential abundance and differential expression pipeline. So when talking transcriptomics, we're talking that we've identified millions of biomolecules, in this case genes. We don't tend to have high missingness, and we have identified biomolecules at the end. We saw for this specific gene in this cancer, we saw this many counts of transcripts, and that's what you start with when you go into the differential abundance pipeline. For proteomics, we're seeing 10,000 to 100,000 generally for the number of biomolecules. So we're several orders of magnitude smaller than transcriptomics already. You tend to have much higher missingness in your data sets. Lots of NAs are not measured. That missingness can be random error. So the instrument might not have picked it up. The identification software that you use might not have been able to identify it with confidence, or it's biologically relevant and you don't really know which one it is. You do end up with identified biomolecules at the end of proteomics. Then on the next magnitude down of size, you end up with FTMS, which you generally have 1,000 to 10,000 identified peaks, varies depending on the specific technology you're using. The missingness tends to be high. There tends to be lots of NAs in these data sets, and we don't have identified biomolecules at the end, which is why I think differential abundance is not commonly used for FTMS, but I'm here to show you that there are things we can do with it, specifically when we get to the class information, that e-meta information that we store. For example, from Daniel's examples, we saw with the Van Crevelen plots, you could get families from each of the groups, like cholesterol, for example, and we can use that information after we get statistically significant peptides, metabolites, or compounds in this case for FTMS. Whew, don't talk about all the omics at once. You'll say the wrong one. And then at the very end of the magnitude of size, we've got GCMS metabolomics. We're talking 100 to 1,000 metabolites identified. There's still high missingness, but we do end up with biomolecules. So here's just kind of all the things you need to think about when you go to do a differential abundance or differential expression pipeline, and how it's variable depending on which type of omics or if you're using FTMS. So here is what we've been doing for this specific example. We identified compounds for the rattlesnake data set in CoreMS. We converted that to Frida. That you could think of that as going from a specific output to a tool to a more general output of Frida. Frida will take output from many different identification tools. And in Frida, we did our pre-processing, we did our quality control, we added e-meta variables for comparisons, and now we can move on to differential abundance, where we apply normalization to FTMS, go through the statistics, and then I like biology, so you're gonna get a little bit of what can I do after I have my differentially expressed peaks. So with the rattlesnake data set, we had two groups. We had our upper control, and then we took some of those from our upper control to the lower area. So that's the upper to lower transplant, and that has a higher temperature, more carbon, and is at a different altitude to sort of simulate as the Earth warms up. What would you expect that upper spots, that upper that upper soil to how would you expect that to change? And so to help aid in statistics, it's very important to think about what your specific question is. And that's what I'm simulating here. And what I'm hoping you guys will do this afternoon is, okay, in arid soil conditions, so how does transplanting soil from an upper site, which is our cooler condition with higher carbon, to a lower site, which is our warmer condition with lower carbon, affect the carbon cycle? And so my hypothesis here is moving that soil to a warmer and less carbon-rich system will stress those microbes. Thus, we expect to see more aerobic respiration in these transplant samples. And then we can measure this as a significant decrease in the abundance of biomolecules containing oxygen. Carbon might not be a big indicator because at the same time these biomolecules are having their aerobic respiration, they might be making other things. But oxygen might be a good way to measure that, because as the organism eats through its carbon stores, it's going to release it into the air as CO2. So first, we're going to get into normalization, how to normalize FTMS data, and I'm going to pass the reins on to Damon here.
Awesome. So yes, so the first question that I think is important to ask when it comes to normalization is just like, why is it needed? And the answer is that it's going to put relative quantities on a comparable scale from sample to sample. When you're running omics data or FTMS data, there's oftentimes going to be a lot of unwanted variability. There could be things like the way that the different samples were stored could be different. They were prepared in a different way, or maybe they were potentially run on different instruments. And all of those different differences between the samples are going to lead to all of this unwanted variability that takes away from our ability to look at the actual biological differences and the questions that we are actually trying to answer with these experiments. So for example, if you look at this figure here, on the left-hand side, we can see this unnormalized data, and the medians of these box plots are kind of all over the place. They're not like centered at the same value, and it shows that there might not be a lot of ability to compare between the samples. However, after they've been normalized on the right-hand side, all of the samples are centered at 0, the medians are about the same value, and we're able to draw more comparisons, seeing which distributions tend to be bigger, which ones tend to be smaller, and make those biological analyses that we want to do. Next slide. And one way that we can do that is using the SPANS algorithm. And this is because no single method works for all data types. There are a plethora of normalization methods, and based off of the data we're working with, there might be a different normalization method that works the best. And so the idea behind the SPANS algorithm is to evaluate those approaches and ensure that normalization is not going to introduce any bias. This means that the highly significant biomolecules are going to remain highly significant after normalization, and the lowly significant biomolecules are going to remain lowly significant. When it comes to how much data is needed, we're going to have at least one non-missing value per treatment group. So if we have 3 treatments, we need to have at least 3 non-missing values for each biomolecule. This was originally developed for proteomics data, but it has been used on FDMS data as well, as shown in Thompson et al. Next slide. So the normalization method, regardless of which one you're going to use, consists of 2 main functions. The first one being the subset function, which looks at, of the biomolecules that we have in the data, how many of them are we using to normalize based off of. So we could be using all of them. We could be using only like 4 of the 7 biomolecules here. And that just, we'll go through that and show all the different subsetting functions and how they choose which ones are being subset. And then the second function is the mathematical function, which is things like the median, the mean, or the z-score, which are the ways that once we find that subset, how do we actually go about the normalization process? Next slide. So for example, if we chose a subset and we found that these were the 7 biomolecules that were being subset and we want to use the median, we would then go through each sample. So for example, this one, sample 1, find the median to be 15. And then in order to normalize, we're going to subtract the median off of those sample 1 abundance values. So biomolecule A goes from 15 minus 15, 0, B 14 minus 15, -1, and so forth. And so we're going to do that for all of the samples, and that's how we're going to get those normalized values, and when using the median centered around 0. Next slide. So then the subsetting functions are going to be these 5 main subsets: all observed peaks, top L order statistics, the percentage of peaks present, the rank invariant peaks, and the rank invariant peaks based off of the percentage of peaks present. And we are going to go in the next couple slides through all of those different subsets to explain specifically what they're all doing. Next slide. So the first one is probably the easiest. It's all. So we're just going to take all observed peaks. So here we have 7 biomolecules, A through G. And regardless of missing values, regardless of anything, we are going to run through all 7 biomolecules being subset and apply the different mathematical functions, whether it be mean, median, or z-score. Next slide. The next method is LOS, or the top L order statistics, which is the proportion L of peaks with the highest intensities. So for example, we could set that parameter to be 0.33. So since we have 7 biomolecules, that means we're looking at roughly the top 2 biomolecules per sample. So for this method, we're going to look at each sample. So we'll start with sample 1, look for the ones that have the highest abundance values, and which in this case is biomolecule E and F at 16 and 17 respectively. And that means we're going to be subsetting E and F, and they're definitely going to be in that subset. We then move to sample 2 and look at the top 2 from that sample and find that it's C and E. And that means those ones as well. So since we already had E, it doesn't change anything, but we're also adding C to the subset. We go through that process for all 6 samples, and any biomolecule that has not been subset is not going to be included, which means that B and D, which never had a peak that was part of the highest third of intensities, are not going to be included within our subset function. Next. Percentage of peaks present is based solely on missingness. So with this one, we were looking at the percentage of peaks present at a specified threshold. So if we set that parameter to be 0.5, we need at least 50% of our samples per biomolecule to have a non-missing value. So going through this system, we're only going to remove biomolecule D from the subset because there are 4 missing values, which makes up 2/3 of the samples. So because it doesn't have enough data that's present, it can't be included in the subset. Next slide. Before I talk about the next ones, we have to do a slight side tangent and talk about the Kruskal-Wallis test, because that's what we're going to use in order to determine the subsetting. So the Kruskal-Wallis test is essentially the non-parametric equivalent to ANOVA. And it's non-parametric because it doesn't assume for normality. So basically, instead of taking the actual abundance values like we see in the top row here, where there are values like 15, 14, 16, we are going to rank them all. So because 13 is the lowest value, that becomes ranked 1, 14, 2, there's a tie for 15, 3, and then 16 becomes ranked 6th place. So instead of comparing the abundance values, we're comparing the ranks. So if those first 3 samples, 15, 14, and 16 were from one group and 13, 15, and 15 were from another, we're now looking to see if the ranks of 3, 2, and 6 are different than the ranks 1, 3, and 3. So the null hypothesis is that the median of all groups are going to be the same, whereas the alternative hypothesis is going to the median that at least one of the groups is different. Next slide. So applying that Kruskal-Wallis test to the subsetting functions, we come to the rank invariant peaks, or RIP function. And this is where we're looking at peaks observed across all samples that have a non-parametric p-value greater than a specified threshold. So here we can specify that to be 0.15, which means that we're looking at p-values that are above 0.15 to be included in the subset, and those that are deemed too significant are not going to be included in the subset. The first thing to note with this one is that missing values are not allowed, which means that biomolecules A, B, and D are immediately excluded from being subset because they have missing value. And then next slide. So then once we apply that Kruskal-Wallis test, doing the ranks for between group 1 and group 2 for biomolecules C, E, F, and G, we find that C, E, and G have p-values that are below that 0.15 threshold, leaving us with only biomolecule F, which had that p-value of 0.487. So as you can see, this may not always be the best option, especially in data sets that have a lot of missingness, as FTMS does, just because here we're only being left with one biomolecule to subset. Next. And that's kind of where the PPP RIP method comes into place, is that it's that RIP method that we just discussed, but it's based off of that percentage of peaks present as well, which does allow us to have some missingness. So for example, if we retain that threshold of 0.5 for PPP, we still have to remove biomolecule D because there's too much missingness, but now we can also consider A and B. So next slide. Next. So we already did the Kruskal-Wallis test for C, E, D, and G, but when we added the Kruskal-Wallis test for biomolecules A and B, we find that those p-values are also above 0.15, which allows us to have that subset of A, B, and F. So we do increase the amount of biomolecules that we're considering when we're using the PPP RIP, rather than simply using the rank invariant peaks. Next slide. And so when it comes to the spans algorithm, you're going to specify which of the different subsetting functions you want to consider and which of the mathematical functions you're going to want to consider. And for each of those combinations, you're going to receive a score between 0 and 1. And a higher value is going to indicate that you're introducing less bias. And so a higher value is going to be the better one. So when we're looking at the actual equation, it's a little confusing, but it's essentially getting at that we're looking at a distribution of the baseline non-significant values as well as the distribution of the highly significant and taking the average between those two distributions. And that's how we're going to arrive at our span score. Oh, I think the slides moved. So when we're using our Rattlesnake Mountain data set, when we apply the spans algorithm, we are going to get that. We're going to find that the Z-score and the RIP method at 0.15 is going to be the best at 0.52. Next slide.
I just wanted to point out, I think your PowerPoint is showing the wrong screen, David. Now we can't see the slides.
David, do you have the slides?
Oh, it looks like
let me start sharing my screen. I have the slides.
Thanks.
Okay, so I usually use Teams. It's not letting me share my screen.
I think we have them too.
Yep, I'm getting them pulled up right now.
Great.
Okay, thank you. Yeah, I have never really had to share my screen with Zoom, so I don't have those systems set up.
Sorry, jump to a random slide though, so you just have to guide me.
No worries. I think we finished that slide. If you go back, yeah, that one.
Sorry, right here.
Yep, you're
Perfect.
good. Thank you. So after we apply then that z-score mathematical function using the RIP method at 0.15, we're able to compare the unnormalized versus the normalized data. And we see that it does a lot better job of putting all of the medians at about a similar place. It's not going to be perfect. As we saw, the best score was 0.52. And so that kind of tells us that it might not be perfect, but out of all the data that we considered and all the span's normalization methods, it is the best one. Next slide. And one thing to really just reiterate is that there is no consistent optimum normalization method. So for example, in Thompson et al., they looked at permafrost data set as well as one related to whiskey. And as we see based off these span algorithm output, it can really range about what's considered the most ideal. For example, with permafrost, it was using the subset function all and the normalization mean, and the mathematical function mean. Or when looking at the whiskey data set, the mean was still the best mathematical function, but PPP at a 0.1 threshold was considered a better subsetting function. So regardless of the omics type or regardless of using FTMS data, you really want to go through that SPAN's algorithm because you could easily find a better normalization method depending on what you're using. Next slide. And so that's kind of what we do with normalization. I think one point to really reiterate is that there's nothing inherently kind of exciting about the normalization. It's not like there are a ton of statistics and analyses to draw from it. It's more that we have to use the normalization so that we can get that data on that comparable scale so that we can actually perform the differential abundance and statistics to identify the biomolecules of interest. Because once we've done the normalization, we can actually get to the more interesting part. And then is David ready for the next slides?
Yeah, Zoom decided I didn't need sound, so I was trying to fix that while taking visual cues from you. So my apologies. We are
No,
fixed.
so then David will take it from here.
We had a demonstration in the flexibility of the teams here at PNNL. All right. So yeah, okay, so I'm taking over share again. Sounds good.
Oh, I'm sorry. I thought that's what you were saying.
Oh, you're good. We're just going with the flow. All right. Perfect. And then share.
Nothing yet.
Yeah, we are getting there. Sure. All right. Present. All right, we back.
Perfect.
Back in business. Okay, so we just talked about normalization. We're going to move on to statistics here. In the PMAR-R package, there is a specific tool for doing both quantitative and qualitative statistics. Because we have lots of missingness, you would want to apply the qualitative tests for cases when there's a lot of absence within the data. For quantitative tests, we're interested in after we've normalized, what are the differences in each group between the mean. And so that would, together with using both that ANOVA test for the difference in the mean and the qualitative G test for presence absence, Bobby Joe et al. Developed the IMD ANOVA, which stands for independence of missing data. And so in cases where you've got a lot of missingness, you can still get more biomolecules return significant than you would get without using this approach. So before applying an IMD ANOVA, you're going to want to filter the data. You're going to need at least one biomolecule in a group for the G test, and you're going to need at least two for the ANOVA. And so that's what this filter is doing here. And in PMAR-R, that function is called the IMD ANOVA filter. For the ANOVA, you want to take an F-test comparison to see if the mean between each of your comparison groups is the same or not. And that's how we get our differential abundance, is after we've determined that the mean is not the same, we can tell you which direction it is, either up or down. So after making that F-test comparison with the alternative hypothesis being that at least one of those means is not equal, then you can compare each of the groups with their log fold change values. Depending on how you normalize the data, that's going to change what these log fold change values look like. So if you've scaled using a log 10 abundance, this is going to be a log 10 fold change. The g-test for missingness is going to determine if the proportion of missing values are associated with a treatment group compared to random chance. And for that, we use Fisher's test of independence with correction for small sample sizes. There are lots of vignettes and instructions online, and since we're low on time, I'm going to just keep this moving. But the main idea here is if you just do the ANOVA test, you might get that, like for our rattlesnake data here, you've got 41 that are positively you have an increase in abundance expression, and then 15 that are down expressed. Whereas when you do the g-test, you get a lot more data, and them pushed together, you get more biomolecules that are differentially expressed. For our rattlesnake data, we had like 8,000, I believe, peaks, and out of those 8,000, we end up with 82 that are actually statistically significant if we use this IMD ANOVA approach. And then as part of this PMR package, we have lots of plotting options for these guys. If you're used to seeing a volcano plot, this is what it looks like, where we've got the fold change on the x-axis, and on the y-axis, we've got the negative log p-value. The idea that, like, if your threshold's at 0.05, it's here, and you could see each of these values, what has been positively or negatively expressed. So we've got that biomolecule information. And just an aside here, as I mentioned before, we've got p-value adjustments available as well. For if you're doing ANOVA or IMD, or if you've got like proteomics data, let's say, you're going to want to p-value adjust for when lots of things come back significant. So now that we've got this statistical comparison, I've got these biomolecules of interest. What can I do with that information? And how does that help us answer our initial question, is what is the difference carbon-wise between our stressed sample from the upper to lower transplant and our lower transplant? But generally speaking, back to omics, there's lots you can do after you've got the specific peaks, or in our case, or generally for other multi-omics, these biomolecules of interest. There's lots you can do because you've separated out the actual biomolecules from the noise, the ones with real change. So you could do correlation across multiple data sets. And this is kind of moving us more towards multi-omics with these general examples here. You can see the correlation of significant biomolecules across these omics types. You've probably seen quite a few networks comparing lipidomics and proteomics and transcriptomics together, of either those being correlation networks or functional enrichment. You can investigate patterns across biomolecule classes, which is what we're going to do in this example. And then even from there, people have been using machine learning once you have more data to say which classes, which biomolecules correspond to each classes for predictions. So if you've got like a cancer phenotype, and I've got these biomolecules, which one of those are more likely to be higher or lower expressed in the cancer case? And then just integrating together all of those data sets is a new and developing field called integration. And that's all of these are sort of examples of integration, but it's very new and in development. So just reminding us of our example, which I just said a minute ago. What can we do? So we've got our differentially expressed peaks. And what I've done is I've just gone back to their e-meta information and plotted the ones that are down expressed or up expressed, and the number of oxygens for each of those peaks. And so the idea is, do we see a decreased expression of oxygen between these two samples? And so you could do a p-value, you could do a Kruskal-Wallis test, which is non-parametric, to see if there's a difference between both of those groups. And you could do the same on the ratios of carbons to oxygens. You don't want to do a t-test here because this is not parametric, it's not normal, it's not continuous. This is count data, and then this is ratio data. So a t-test is not appropriate in this case, but you can use Kruskal-Wallis to see if there's a difference between those two groups. So we see a significant decrease in the expression of biomolecules with high oxygen counts and OC ratios, which does suggest that in these stress samples, we're seeing more carbon metabolism and it's being released into the air as CO2. You could, from there, then take that information of what's been differentially expressed and do a KEGG analysis. There is a layer of uncertainty you're adding because we don't know exactly what the true biomolecule is for the formulas that we have. But just running this through KEGG, I see an increase in cellular defense activities. We're seeing more macrolide synthesis and wax biosynthesis and xenobiotics. So the bacteria in these samples are producing or gearing up for a more stressed environment. And then we see a down expression of like arachidonic acid metabolism. You could kind of see a shift in the microbes towards protection, less biosynthesis in general. So that's what you can do with KEGG with this data at this point. There are quite a few limitations though to this approach. Not all the biomolecules are known. Not all the biomolecules from KEGG have pathways. One molecular formula can map up to 10 biomolecules. You can have not enough significant biomolecules to even do this, but you can kind of get a snapshot from FTMS of what might be happening biologically. So now that we've answered our question that we know that they're stressed, what is happening on that level and what kind of omics do I want to do from here from FTMS? Maybe I do want to do metabolomics at this point, more targeted towards these areas like GCMS or LCMS, or maybe this is lipidomics here. So we answered a question, and we're proposing our next question with the data we had before. So just a high level overview of the pipeline, once again, from formatting data to normalization and kind of reviewing all the things I mentioned about. Though there's a general step-by-step procedure for differential abundance and expression, the exact omics and technology that used will change how each of those steps are conducted. FTMS does not have biomolecule identifiers yet. So it is not traditionally what we think of in biostats as an omics type. We can still conduct our pipeline if we're asking a question that can be answered with differential abundance, which is what we did here. This pipeline at the end produces those significantly changed biomolecules for comparison with an indication of whether the abundance expression is up or down. And then from there, it is back to us to interpret what those results mean biologically, whether you use something like KEGG or you compare those E-meta groups, or if you've got multiple omics, you try to put them together in networks. The point is to then take what we got from the statistics, from the noise of the instruments, from the missingness in the instruments. Now we have our snapshot answer. What do we do with it now to move forward? So what questions will you answer? Here's your data. We're going to do your seminar here in like an hour. So get thinking about it. We're here to help you. And I think too, it's always important to remember that all of these tools are built on all of these other scientists who have done so much work. This is just a snapshot of one of the many people that who have developed the FTMS and PMART tool for us to use in our analyses. So happy to answer any questions at this point.
Thanks, David. That was great. Damon, you're on the Discord, right? Can you see Monica and Chelsea's questions?
I see Paula's question. For the KEGG analyses, are you only using biomolecules that are confident on the formulae? Yes, and that was done way back in the core MS to Frida step, where Natalie only returned molecular formulas where we were confident that they were assigned correctly. I see several people are typing, so.
Yeah, there was, so there were a couple that got answered in the Discord, but from Monica Schmoll, why filter out molecules in only one group? This might be most informative just because they are unique there.
One more time. Oh, I see. Okay, why filter out molecules in only one group? What is this referring to specifically? Is that normalization methods?
We'll see if Monica's still on to.
Oh, I see. So when she's asking this question, she's referring to the filtering we do before the statistical analysis step. And that's because you can't do the stats test without enough data. So that specific filter is to remove cases where we just can't perform statistics on it. We try to make up for that with the G test, with the independence of missing data part of the IMD and NOVA, but you still have to meet a minimum threshold of measurements, yes. Ooh, batch effect question. All right, so there are always some batch effects in omics data. Normalization can remove some of them, but probably not good enough. What tools would you suggest to evaluate and or eliminate batch effects to some extent? There is a companion package to PMART-R right now being developed called Dancer, which has a whole bunch of normalization methods for batch effects for the different omics types in development. So I would recommend what we finish putting in there. I can send you more specific details. I am not on that project, so.
There was one from Chelsea St. Germain that was touched on by Will, but this may have been mentioned over the last couple of days, but what is the mass range for FTMS instruments? Is the range large enough to detect a lot of the RNA transcripts and proteins in the sample, or is it more targeted at smaller masses? They are all +1 charge molecules, right?
Yeah, so you could do some FTICR with Orbitrap, it looks like, for intact proteins, but for these specific examples, we've limited our range from 200 to 900 for the data off of these instruments. So for our specific example, you wouldn't be catching many intact transcripts, but you could be catching small protein fragments with that. Will is probably better for that since he runs the instruments. Yeah, so Paula brings up a great question too. We've been discussing how FTICR MS data is not very quantitative, but so do you think it's okay to use the data for differential abundance analysis then? If you are not confident in the quantitative part of your data, you can just run the G test, which is a qualitative test for differential abundance, to see if you get something that is useful. A lot of times we don't have endless time, but you might want to adjust your comparison groups if you have too many, or think about how your question needs to be answered differently. Or maybe there's just not enough data or enough significance in your data for that to be useful. So yeah. Oh, Damon's answering a little bit more about those batch effects correction methods.
We have one from Rachel Berner. How amenable is this pipeline to multivariate analyses, such as adding in soil nutrient data, vegetation community data, et cetera, to test for environmental factors that may be driving the metabolic response you'll see in soil?
If you can add those variables to meta information about each biomolecule or about each sample, you can creatively answer those questions. But without seeing this exact data and how it's different from what I'm used to seeing, I don't know exactly how it would fit in. But creativity is key sometimes. You know, as long as you don't violate the rules of statistics, which is the point of developing a package to help you not do that as you do these analyses, it's kind of hard to mess up. Even like back to normalization, we mentioned there were many normalization methods you can use. There's really not such a thing as an invalid normalization method. Rather, it's a normalization method that introduces bias into your data. That's what you're trying to avoid with the test. But if you've got a few that are similar performing, or as long as they don't fail that Kruskal-Wallis test that they've introduced bias, any of those is okay. It's statistically, it doesn't violate the rules of statistics. So